right, so we are recording. All right, Brittany, are you all set?
Yeah, I'm good to go.
Okay, so Brittany is joining us tonight from Save Barnegat Bay. She is their Jellyfish Education Coordinator, and she's going to talk all about jellyfish in Barnegat Bay and the Stop the Sting project that they have. Brittany, I'll let you take it away.
Hello, everybody. Like Caitlin said, my name is Brittany, and I am the Jellyfish Education Coordinator here at Save Barnegat Bay. And we are going to be talking about our Stop the Sting program. So let me just share my screen real quick. I have a little slideshow for everybody. Okay, perfect. And then everyone can see, can everyone see?
Yep, looks great.
Perfect. Okay, awesome. So Stop the Sting, we are working in partnership with a couple of different nonprofit groups and organizations. So we have the Berkeley dive team. It's Berkeley Township Underwater Search and Rescue Squad. It's an all-volunteer dive team. They will be doing most of the scrubbing that we're talking about today. We have the MATES program, the Marine Academy for Technology and Environmental Science. We work with students and Dr. John Wennick from MATES. It's a high school located in Manahawkin. We also have our jellyfish scientist, Dr. Paul Bologna, works at Montclair State University. He is doing most of the research for this project that we're doing, and this program is funded partially by the NJDEP, Department of Environmental Protection. So with that, away we go. So before we talk about what we are doing, just a quick where are you, where we're working. So this is the Barnegat Bay watershed. I'm sure most of you are, or if not all of you are familiar. We will be focusing on the northern part of Barnegat Bay. So kind of this cutoff at Oyster Creek almost is kind of the natural cutoff for the type of jellyfish that can be found and the type of jellyfish that we're focusing on for this project specifically. So in the northern bay, there are a higher concentration of bay nettle jellyfish, which are the stinging jellyfish that we're referring to when we're working and talking about this project. And then on the southern half of Barnegat Bay, the most prominent jellyfish is the comb jelly, which technically is not even a jellyfish, but they are related and they're very similar. So that's kind of just the cutoff section, and that's why we're focusing most of our work with the bay nettle jellyfish in the northern half of Barnegat Bay. So then Stop the Sting. What is it necessarily? We are trying to reduce the amount of bay nettle jellyfish in lagoons along Barnegat Bay. We have certain targeted areas that we'll be focusing on, and we'll be doing this project and this study for 3 years. Why? Because jellyfish are taking over our waterways and harming our native critters and changing our lagoons, making it more difficult for us to enjoy our bay. So they are kind of a nuisance. People are finding it harder to swim, and they're kind of everywhere. Every single time you jump into the lagoon in their backyard, you see at least one bay nettle jellyfish. So it's not really as safe for people to be swimming in these bodies of water anymore, which is kind of stinks, obviously, because then we can enjoy lagoons, we can enjoy our bay, and it's not as fun of a summer if we can't enjoy the things that we love the most about where we live. And then how are we doing this? So we will be scrubbing bulkheads and floating docks and studying the bay nettle populations throughout these next 3 years. And we'll talk more about that in a minute. So I keep saying bay nettle, but we don't really know what it is. This is the bay nettle jellyfish. They are very closely related to a sea nettle. So sea nettles are found in the Atlantic Ocean, all along the Atlantic Ocean. They are very, very similar in look and size. The bay nettle is a little bit smaller. Its scientific name is Chrysaora chesapeakei. It's kind of hard to pronounce. And the size, usually the bell can grow up to about 7 inches in width. And their diet is mostly small critters, worms, fish eggs, shrimp, baby crabs, and even other jellies. So I talked about the comb jelly before. They also eat the comb jelly, which is why comb jellies aren't found as predominantly in the northern part of Barnegat Bay. Since the bay nettle has kind of taken over that area, there are less comb jellies because they are in competition.
Hey, Brittany.
Yeah.
There's a gray box. Yeah, it might be like a drop down, something that's blocking the screen at the top. Yeah, right where like your cursor is.
Yeah.
Is there a way to like move that, or because it's blocking some of your words on your presentation?
Yeah, I'm trying to I can't really get rid of it.
Okay.
I'll put it at the bottom because there's usually not really words or anything in this little part.
Okay, there you go.
Or right here. Okay.
Okay,
yeah.
thank you.
Sorry about that. But yes, where was I? So up until 2017, actually, we did not know that the bay nettle was a specific type of jellyfish. We just assumed that any white bell jellyfish that looks like that was the sea nettle. But it was discovered that they are actually genetically different. Their DNA is slightly different, which makes it a completely new species of jellyfish. And it was actually discovered in the Chesapeake Bay, hence the scientific name Chesapeake Eye. Little fun fact. And then to know how scrubbing bulkheads and such, we have to look at the life cycle of the bay nettle jellyfish. So during our scrubbing process, we are focusing on the polyp stage of these jellyfish. So this circled area right here. The bay nettle jellyfish goes through two different forms of reproduction. So we have, as you can see at the top, the adult jellyfish, and we have the sexual reproduction with the fertilized egg. That turns into this planula right here, which kind of just looks like a furry little tic tac, kind of moves around the water with these little arms that surround that little egg-shaped organism. And it kind of just swims around and looks for a hard surface to attach to. So then that's the attached planula, then eventually grows and forms into a polyp. This is where the second stage of reproduction starts. So this polyp can either copy itself from side to side, or also start the budding process. So it'll start making kind of stacks of pancake-like looking organisms, jellyfish eventually, and it kind of just stacks vertically up and down, and or it can make copies of the polyp, the actual polyp, from side to side. So this kind of happens during the fall and winter when we don't see the adult jellyfish anymore. They have the polyps going through their copying stage, their second stage of reproduction, forming this budding polyp. So you kind of see, if you look under a microscope, they're hard to see with the naked eye. You'll see little tiny stacks of just really long chain of budding bay nettle, eventually bay nettle polyps. And then as the water gets warmer, those budding polyps start to detach, and that's the ephyrae. So it's like the baby jellies, what I call it, just because that's exactly what it is. It's just a juvenile adolescent jellyfish that starts swimming around. They're kind of hard to see still because they are so tiny, and then they eventually grow into the adult jellyfish you see, and then the life cycle starts and begins again. So that's kind of a good little demonstration to show what is happening. I'm going to move this out of the way again and put this at the top right here. And then this is kind of what they look like realistically. So we have the polyp goes through metamorphosis and their cloning stage, and you could see the stacks kind of growing and getting bigger and more mature until eventually they pop off into little small baby jellies. Okay, so why are these jellyfish such a problem right now in lagoons? We saw them pop up and become a big problem in the Chesapeake Bay. Now they're becoming a big problem in Barnegat Bay. Why is that? And why are lagoons the places we're finding them the most? That is from a changing ecosystem. So as I said, these polyps have to grow on hard surfaces, and that's where the reproduction starts and begins, on these hard surfaces. If you're looking at a natural edge, an untouched body of water, there are hard surfaces. We have rocks, we have shells, we have a bunch of places where polyps could attach, but they're kind of few and far in between. And where they exist, they're not very large. So if we look at lagoons now, the whole entire ecosystem is completely lined with hard surfaces. And we found that these bay nettle polyps prefer vinyl bulkheading. So these vinyl bulkheading is good because it lasts longer, and chemicals, we haven't found that chemicals are really leaching into the bay as in the treated, unlike the treated wooden bulkheads. But that provides a huge habitat now for all of these polyps. They can continue cloning themselves from side to side. They can continue cloning, stacking, making little baby jellyfish stacks for hundreds and hundreds of miles of bulkhead. So that obviously causes a problem. And since it's harder for other things, settling organisms, to attach on these surfaces, it kind of eliminates competition as well. So if we don't find a ton of barnacles or anemones that can attach to a vinyl bulkhead or to a plastic floating dock, then there's no competition for these bay nettle polyps. And they have more space to grow. So just to visualize that again, we have these same areas. So you see a jellyfish right over here, few and far in between, again, because they're competing for very little amount of hard surfaces. But then we see all of these polyps starting to form and plenty of jellyfish in this area. So what are we doing? What have we been up to? We have our volunteer divers from, like I said before, Berkeley Township Underwater Search and Rescue, have been out all fall long and all spring long and into the summer still. We haven't finished yet. Scrubbing bulkheads and the underside of floating docks in the Berkeley Shores community. So we had people, we sent out a mailer to everyone living in Berkeley Shores, offering a free scrubbing program, telling them that we are trying to reduce the jellyfish population by scrubbing their bulkheads, and the program is completely free. We will go out and do it. You just have to sign this form. And we've had about 100 plus homes sign up for the program so far. And yeah, it's pretty good. I'll show you the map right now. So you kind of, oh, just kidding. You'll show you the map soon, but we will be scrubbing bulkheads. As you can see, this is Grace Ann, Save Barnegat Bay's education coordinator. She's also on the dive team. She's also a volunteer on the dive team. And we are also scrubbing floating docks. So the underside of floating docks, jellyfish prefer when they do the stacking cloning. It's kind of, they are found they like to hang upside down as opposed to growing out from the side. So floating docks can house up to 100,000 jellyfish polyps, we found. So scrubbing the underside of floating docks or taking them out in the off season and then putting them back in, reinstalling them during that short summer season could actually greatly impact the bay nettle jellyfish population in the coming summer years. And then this is the first place we've been scrubbing. As I mentioned before, this is Berkeley Shores. All of these little things, we have these flags. That means that their house, the properties were scrubbed. It was a successful. All these little targeted areas are places we have left to do. Right now, these little blue signs mean they signed the form. The other targeted areas are just that they signed up, but they never completed the application process. So we have about 17 homes right now that signed up. They're all good to go, and they just need to be scrubbed. We will be finishing that hopefully this month, and that would complete our whole entire first year of this experimental scrubbing program. So we have these scrubbers come in from the dive team. We had them come in last fall for about 2 or 3 weeks. And then we've been working all spring long, every Saturday or every other Saturday, going out, scrubbing bulkheads, scrubbing floating docks. And then we are going to have our scientist, Dr. Paul Bologna, go back in and do polls. So we have toe polls where he's trying to measure the amount of DNA, jellyfish DNA in particular, in the lagoon. And we're also doing dock swabs to measure the jellyfish DNA on docks and bulkheads who have been scrubbed. So he's going to compare the data between the bulkheads who were not scrubbed back last summer before we started the scrubbing program. He has samples of docks and floating docks before they were scrubbed, and now he's going to go back in this summer and swab docks and bulkheads that were scrubbed to see if we could find a reduction in jellyfish DNA in these areas. And if we do, that means that our process is actually working. So that's exciting. And here's just more pictures of the divers scrubbing. They're kind of fun to look at. Thought I wanted to show you. We use a power washer to scrub vinyl bulkheads. It's just quicker that way, and vinyl bulkheads are more sturdy, so it's safer that way as well. And then, as you saw in the previous picture, we just use pool brushes to scrub wooden bulkheads and to scrub underneath the floating docks if we can't get to it with the power washer. This is actually a jellyfish. This is a lion's mane jellyfish that we found while we were scrubbing. We thought it would make a cool picture. It's not actually a bay nettle jellyfish. It was too cold to see any bay nettles out in the water yet, but still cool picture. Our floating dock again. Our diver just chilling, waiting in the water, taking a quick break. And then this is our other diver. So we have two different boats that we use. He has all his dive gear on because he is an inspector. So he will go in and inspect each signed up home to make sure there's no structural damage to the bulkhead that we can find before we go in and kind of mess with it a little bit by scrubbing with brushes or with the power washer. If he is concerned about any structural damage or possible structural damage, he will fail the house for inspection. So that just means that we don't want to. We warn the homeowner that we're concerned about the integrity of their bulkhead, and we suggest that they get it checked out by a professional because we are not professionals. We don't know what a bulkhead should look like, have a pretty good idea, but we suggest to get it looked at by a professional, and we don't scrub their property. We opt out of scrubbing for this season. But if they get their bulkhead fixed and we're back out in the next summer season, we will absolutely re-inspect them and scrub their property if we think it's safe. I think I saw a question. Okay, so we have a question from B. Hunter. For your divers, are they allowed to use air tanks, or does it have to be free diving? They can use air tanks. Usually when we're scrubbing, they can just walk along the bulkhead without really being underwater. So we have the air tank for the investigator, the inspector. We have him or her in full dive gear with an air tank because they have to get and actually look at the whole entire bulkhead. So they are underwater for a majority of the inspection. For the people scrubbing, I'll go back. So for the people scrubbing, you kind of see the free diver. They can wear gear if they want to, they just choose not to. They're standing along the bulkhead and kind of just taking the brush and just pushing it down, up and down, and just lightly brushing over the bulkhead. So they don't really need it, I guess, but they can if they want. The inspector is usually the one with the air tank and full gear. Of course. Okay. I also have some videos of us scrubbing so you kind of see what it looks like. And in the video, one of the videos, you'll be able to see exactly what I mean, that they're just usually can just stand in the water. Let me see. 1 second. Videos are not showing up. Okay, we're gonna do it this way instead. 1 minute.
While Brittany gets her video set up, if anyone has any other questions, feel free to put them in the chat now and until we are ready to show. Oh, it looks pretty good, Brittany. Yeah.
Perfect. So this is a video of Grace Ann scrubbing a bulkhead. you can see she just takes the scrub brush and just kind of pushes down. And we're doing this lightly because there are other living things that are filter feeders and settling organisms that are good for our bay and help create a healthier bay. So we don't want to take everything off. It's pretty easy to detach a jellyfish polyp from the bulkhead or from the floating dock. So we don't want to cause major issues or major damage to other living things that are living on these bulkheads. And then we also have this video of our other volunteer, Lauren, using the power washer on a floating dock. This one's obviously a bit more powerful, so it'll take off some more things. But jellyfish polyps are the main thing that kind of grow underneath the floating dock, besides barnacles, which are obviously very hard to remove.
Brittany, I have a question actually. Is this something that residents can do themselves, or do you think it's not safe for folks to try to do this on their own? I know you probably have their own way of protocols, but I was just curious, people can try it at their own properties.
Yeah, we've actually been talking to a few people, and I would suggest you can do it on your own. We would just suggest, again, just using a pool brush and lightly scrubbing against your bulkhead. We don't want anybody, if they have an old bulkhead that's slightly damaged, that has maybe some holes, a few holes in it, we don't want to cause a problem where they're further damaging the integrity of their bulkhead, obviously. But it's a simple pool brush, and you're just lightly going over it once or twice. Again, we're just trying to detach the jellyfish polyp, the bay nettle jellyfish polyp. So a few brushes over your bulkhead lightly should do the trick. And then once they're detached, they cannot reattach, and then they can't clone themselves and make copies. So you can, short answer.
Okay, great, thanks.
Of course. And then one last one, you can see they're in like hip high water, just going along a property, a very long property, with scrub brushes. Okay. This out of the way again. Okay. Perfect. And then more pictures. So this actually, the guy with the little headset, his name is Greg. He's the chief of the dive team. He's chief of operations. And the guy with the sweatshirt, his name is Carl. He is the president of Berkeley's dive team. So Carl and I usually work together from the dock and talk to homeowners and do all of the outreach and explain what we're doing and why we're out, because plenty of people come out, they hear the loud boats and they're like, what's going on? What the search and rescue squad, did someone lose something? And we just talk more about our jellyfish program. And usually we get more signups as we go, which is also fun. And then while everyone's doing all the hard work and getting down in the water, I'm just standing there, mostly watching them, making sure everyone's okay. My friend came with us and took pictures. It was nice. Okay, so that is one major aspect of our work and what we've been doing. We also have a community science program. So while we're only scrubbing in targeted areas, the community science program is open to everybody living along Barnegat Bay. So what we're asking of people is to hang settling plates. So there's these little tiny blue plastic squares that are suspended on fishing line. And they act kind of as a temporary habitat for not only jellyfish polyps, but other settling organisms that are typically found in our lagoon communities. So we have volunteers come in, pick up a set of plates, some instructions that come with them, and we have them just hang over their dock or their piling into the lagoon. After a couple months, we invite them back. So wait, hold on. Actually, this is what they look like, kind of. Just simply like that, just as long as the plates are in the water, if not fully submerged, at least partially submerged. We have them sitting in the water for a couple of months, and then we invite our volunteers back to what we call, oh, and here's what they look like. You can see them. Little settling plates in the wild. See, you can see like the little top first plate, but the rest are all mostly submerged. And then after we invite them back to what we call an observation day. So when observation day comes around, they take their plates out of the water, bring them in a bucket of lagoon water, and they work with scientists like Dr. John Winnick right here, or student scientists from Mates or from Montclair State University that work with Dr. Paul Bologna. And they do surveys to see what is settling on these plates. So you get to sit with the actual scientist or student scientist and go through their survey. So they're documenting the percent of coverage of each settling plate and what's sitting on the settling plate, what's settling there, how much of the settling plate is covered with that, on which side of the settling plate. It's pretty cool, and you get a whole description on different kind of things that are growing in your lagoon that you aren't usually able to see on the everyday. Here's Grace Ann again, looking at, I think this was our first observation day. There was actually a jellyfish polyp that we found, which was super exciting. So the community science program was working. And it's nice because it provides, we can't use this research and what we found, our findings, in our actual paper and our report, because these settling plates are not to a specific standard, but it does give us information on where we should look. So if we see a lot of jellyfish polyps popping up in Berkeley shores, as we expected, then that means we are hitting the proper targeted area. And then if we have settling plates all over Barnegat Bay, that can kind of tell us where we should focus our efforts next. So we're planning to go in Toms River next, in Green Island, because there's been documentation of jellyfish polyps there. But if Beach Haven West also has a high number of bay nettle jellyfish. They started moving south a little bit more. If we had a high concentration of community scientists in Beach Haven West, and we were found that there's been a lot of bay nettle jellyfish polyps on these settling plates, maybe we could transition our work to that area. Maybe that should be our new targeted area in our next span of research when we're trying to figure out where to move on to next. So while we can't use all these findings scientifically, we can use them to judge where we're moving next, where we're doing more scientific research, which is also really cool. And we get to see, everyday people get to see everything that's growing in their lagoon, which is also very fun to do. And then here's a student scientist doing a survey. You can see all the greenery. And then, let me move this out of the way. Sorry, this little thing is a little annoying. But yeah, so you're surveying what they found on each plate and documenting it. So what are some things besides jellyfish polyps that we see on these plates? I'm not going to get out of the way, that's just the title. So what we've seen, that was from our virtual observation day back in January. Here's a few things of what we saw. This is a sea squirt colony. Mostly when people see these, they think they're jellyfish eggs, which is not the case. We know that if they're settling, they're jellyfish polyps and they look very different. They're kind of cool if you squish them the right way. They kind of squirt water back at you. That's why they're called sea squirts. You can see a barnacle right here, and then we have the two worms coming out from underneath everything right there. So as you can see, the settling plate came from Berkeley shores. It's almost completely covered, and it's only been in the water. I think it's only in the water for 3 months tops. So it's really cool how quickly these organisms find a place to settle on and just attach themselves. And then these are jellyfish polyps that Dr. Paul Bologna had in his lab. It's in a petri dish, so it's not really attached to anything that we could see. But this is kind of what they look like in real life. They're kind of harder to see and capture. This is what they are through a microscope. And this is our jellyfish scientist, Dr. Paul Bologna, holding a bay nettle jellyfish. So as you can see, move this out of the way a little bit so you can get a look. So the bell, he's holding the bottom of the bell and getting stung all on his hand, but he says he's used to getting stung, so it doesn't bother him. And you can kind of see how long the oral arms actually are and the stinging oral arms are. The bell is pretty small. Again, that's only about 7 inches, but this one might have been even 10 inches. But it has a pretty small bell in comparison to the rest of its body. This was a couple of feet long. It was very, very cool. And this is just another view of our first observation day. So you can kind of see in the back, we also had student scientists who did research projects related to the jellyfish program that we're doing, presenting their work. We had the microscopes all set up with scientists, student scientists manning them and documenting settling organisms, and then just more information about our project specifically for Berkeley Shores members. This was at the Berkeley Township Underwater Search and Rescue's dive house, where they set up shop. We had Berkeley Shores community members come in so they can meet the dive team, get to know what they do a little bit more. We had Dr. Paul Bologna give a talk about jellyfish. And we had a couple of settling plates come in for inspection, which was really cool. And then another research aspect of what we're doing, we have a student grant program. So the student grant program has been run through Save Barnegat Bay for many, many years. We have a water quality team and some independent research teams throughout the past 2 years dedicated to specifically jellyfish, but the student grant program and water quality, usually they focus on water quality issues. But they also do independent research and they do wildlife surveys, and that program's been going on for years. We have just a couple that are dedicated just now to our jellyfish program. So what the water quality team actually looks for, so they do testing every week. They test for water temp, salinity, pH, dissolved oxygen, turbidity, suspended solids, E. coli, conductance, and conductivity. So I'll go into a little bit more what some of these mean. So for water temp, bay nettles have been found in higher concentrations in warmer waters, and water temperature and dissolved oxygen levels are inversely related, which means as water temperature rises, the dissolved oxygen level typically falls. And then we have salinity. Bay nettles have been found in areas where there's little change in salinity, usually in lagoons. There's not a drastic change in salinity content, or salt content, which is another reason why they would prefer lagoons. And then we have dissolved oxygen. So dissolved oxygen is simply the amount of oxygen that is dissolved in a body of water. Bay nettles don't necessarily prefer less oxygen in the water. They just can survive it. They're very adaptable. So they don't need a high amount of oxygen to survive, while other native organisms, fish and juvenile organisms do, if that makes sense. So there's less competition because these native animals who have thrived in Barnegat Bay are used to a certain standard of water quality. And now that the water quality has been decreasing, they have a harder time surviving, and the bay nettles can just beat them out. Turbidity is water visibility. So usually due to the amount of minerals or individual particles in a body of water. We have suspended solids, which measures particles in the water samples. E. coli is just the bacteria survey that they do. Conductance is the degree to which an object conducts electricity, which is closely related, obviously, to conductivity, which is the measurement to determine water's ability to conduct electricity. So it's an accurate and inexpensive way to measure the ionic compounds in water specifically. I know that's all a lot and very science-based, so if you have any questions, I am not a scientist, but I can try to more easily explain it if need be. Just let me know. So now we're going to go to results from year one. So last year, we had 3 different projects focused on jellyfish from the student grant program. Project 1 was an analysis of zooplankton populations within Barnegat Bay. And his conclusion was, his name is Anthony, his conclusions found that bay nettle populations do impact zooplankton populations. And as bay nettle populations increase, it could potentially have disrupt the natural food webs that are in place in Barnegat Bay or have been in place in Barnegat Bay. We have a second project, which bay nettles and lagoons and assessment of water quality in Berkeley Shores and in Silver Bay. Their conclusions were that Berkeley Shores has the lowest mean level of dissolved oxygen and little variation in salinity. So Berkeley Shores should in fact remain a targeted area for our study in the next 3 years. And then the third project was documenting the settlement of Chrysaora, Chesapeakei, and other organisms on vinyl bulkheads in Barnegat Bay. This person was Lauren. She found that more research over a longer period of time should be done to come to a more definitive conclusion. She could not come to definitive conclusions with the amount of time that she had. She only, these student scientists only have a couple of months in the summer to work on their projects. So she established that more time needed to be done and dedicated to come up with an actual conclusion for the project. Any questions on that? So our student science, student grant program, we have students come in over the summer. They either apply with independent research proposals, or they join a water quality team or a wildlife survey team, and they actually get paid for the work they do, which is cool. We get to have actual student scientists do actual science research that can be published officially as a report and as a study along Barnegat Bay, which is also very cool. And you could find all of the student science projects and student grant projects from all of the years that the program has been in place on our website. If anyone's interested, I can send you a link to any of those, which you would find these projects on as well. So you get to learn more about the science that they've done for Jellyfish Project. What's next? So I talked about how we worked or have been working in Berkeley Shores for all of the first year, starting last May. We are now moving on to Green Island, specifically in Toms River, specifically this little area of Green Island. So it's about 6 roads, 6.5 streets lined in lagoons. We're trying to figure out, or we picked a smaller targeted area because we have only one volunteer dive team with only a certain amount of volunteers who are available to scrub when we do the scrubbing. So we have to go back to Berkeley Shores, hopefully next spring. That's our goal. But we want to be able to do that, we need to finish Green Island Toms River this fall. So we picked a smaller targeted area. Hopefully we can get higher concentration in a smaller area of signups, which would be better for our study because we can get every street to have a majority or at least half of the houses or bulkheads scrubbed. There's a better chance that we'll get better results when we're doing the dock swabs and the polls from Dr. Paul Bologna. Okay, so last but not least, how you can stop the sting. So everyone has can do their part to help reduce the jellyfish problem, no matter if you're living on the water or not. Some things, if you are living on the water, if you can do. If you or someone you know lives in the targeted areas, so Berkeley Shores or that section of Green Island, you can sign up today to get your bulkhead scrubbed. You can sign up for the next couple weeks to get your bulkhead scrubbed or your floating dock scrubbed. Take any floating docks. If you're not in the targeted area, you could still help by taking out your floating docks or any other potential settling habitats. Take them out during the off season, and you can reattach and reinstall them in the summer season when they're in use. If they're only going to be in use for about 3 months out of the year, there's no need to have them in the water collecting all those jellyfish polyps for the majority of the season or majority of the year. Also open to everybody, you can join our community science program. So pick up settling plates, pick up a set of settling plates, get on our email list, and come back to our observation days and get a look at what's been settling in your lagoon, what typically settles in your lagoon. It's a very fun program. It's good for kids who are really excited about science and looking at critters. It's cool for adults to actually be able to see what's actually growing on their bulkheads, because let's face it, no one's really interrogating their own bulkhead, looking at what's typically growing on there. It's kind of fun to actually be able to see it in person with a microscope and having a scientist explain the function of these settling organisms. And then also, as we already discussed, you can scrub your own bulkhead gently using a gentle bristled brush, pool brush specifically. You can just go over your bulkhead a couple of times and that should do the trick. But even if you, hold on, move this out of the way again. Everyone can help. So even if you're not living on the water, you still have things that you can do to help reduce the jellyfish population. Limit your use of fertilizer. So the reason why, one reason why bay nettles are so prominent in our waters now is because a decrease in water quality. So since these jellyfish are very adaptable, they're not very picky with where they can live, they can survive in poor water quality conditions, while other organisms that often compete with them cannot. So if there is less competition due to poor water quality, increasing water quality and improving water quality would create more competition and naturally lead to less bay nettle jellyfish. So this is where everyone else comes in that's living in our watershed. If you limit your use of fertilizer, you can take the Barnegat Bay Fertilizer Challenge. That would decrease the amount of nutrients that is getting into our bodies of water, improving water quality overall. One thing you can do. You can limit runoff, so catch it and soak it is kind of our terminology. We have a charting a course for Barnegat Bay Stormwater Series with stormwater specialists and experts, Chris Abrupta and Steve Souza, I think his name is. One series, it's over Zoom. One series is already took place, I think it was June 21st, but we have the whole entire session recorded and on our website. I can show you that. And our next session will be July 21st, which I think Caitlin mentioned that was the drift Q&A, if I'm not mistaken. But that will also be recorded anyway and put up on our website. So get to learn more about stormwater and runoff and how you can help eliminate or improve our runoff that's entering Barnegat Bay. So one way to catch it is our rain barrel. So if you install a rain barrel, we have rain barrel workshops. Grace Ann is working with Rutgers actually, and for our non-point source pollution grant to do a bunch of rain barrel workshops all over the watershed. She teaches you, supplies you with a rain barrel, teaches you how to put it together and install it. And then that catches rainwater that's falling off of your roof so it doesn't fall into our storm drains. Once it's captured, it's stored there. You can use that to slowly reintroduce that rainwater back into the environment. So when you're watering your garden or washing off your car or just draining it slowly, if you don't have a garden, if you don't have anywhere to put it, if you're just slowly draining it back into your lawn, that is eliminating the amount of runoff. It's like a 55-gallon drum, so 55 gallons of water then would be reintroduced to the environment without running off into our roadways and then eventually into our bay and picking up all of the fertilizer and everything else that comes along with runoff. Soak it, like I said before, you can water your plants, your gardens, or if you have a native plant garden, we have some resources to help you create your own native plant garden. Native plants obviously soak up water much better than the typical grasslands that we have now, because they are made for this, sorry, that's our ring. They are made for this environment, so they are better suited for this area, meaning they can capture more water, they can drain it better. That also reduces the amount of runoff that's entering our bay if we have native plant gardens. And last but not least, you can advocate. So talk to your community leaders, your council members, mayors, state representatives, and even governor more about their stormwater management system and their plan and what they can do or what they are already doing to improve it. What are their plans to improve how we manage our stormwater? Right now, our stormwater just goes to storm drains and eventually just goes right into Barnegat Bay. It's not filtered out, it's not preserved. There is no process to make it more clean before it enters our bay, which obviously decreases the water quality of Barnegat Bay. So what is our government's plan to counteract that issue? How are we going to create better stormwater management systems to improve water quality for Barnegat Bay? What else you can do is join our captains program. So you can become a Barnegat Bay advocate. Captains just go to their local municipality meetings. So their monthly meetings, land use meetings, environmental meetings, anything. They take some notes and they come back to 1 month. We have a monthly meeting, I think, on Fridays, where all of our captains come together and just discuss what was talked about at their municipal meetings. You can sign up through that through our website as well. And then finally, contact us. So if you ever have any more questions, you want to get involved in the Jellyfish program specifically, again, you just look for me. My name is Brittany McLaughlin. My email is jellyfish@savebarnegatbay.org, very easy to remember. Or you can check out our website at savebarnegatbay.org/educate/jellyfish. If you want to learn about other educational programs like Grace Ann's Rain Barrel workshops, you can contact her directly. Her name is Grace Ann Taylor, and her email is educate@savebarnegatbay.org, or you can go to the website savebarnegatbay.org. I think it's /programs now, not educate. I think it was just changed. But I could link, I'm going to send the links in the chat anyway, so you'll get the direct link to that. And then if you want to advocate, if you want to learn about more about our drift screenings, or we have drift podcasts and the other programs that necessarily aren't educational, you could talk to our executive assistant and policy coordinator, Avery Lentini. Her email is assistant@savebarnegatbay.org. And the link to our website for advocacy is savebarnegatbay.org/participate. And then that's all I have for our slideshow. I could also show you the links to the website, and I could put the links in the chat. But yeah, that's it. Do we have any questions or anything? I know I kind of went through that a little bit quickly.
Well, thanks so much, Brittany. That was great info and sounds like a really great project and getting community members involved and students too. So thank you for sharing. Yeah, please do share any links that you'd like in the chat. And if anyone has any questions, feel free to come off mute or put your question in the chat too, whichever one you're more comfortable with. I just want to give you a remote or virtual round of applause, Brittany. So if anyone wants to use any of your reactions, go for it. Thank you again.
Thank you.
But
Thank you.
yeah, absolutely. Any questions right now? There's a question in the chat. Are there any other types of jellies that you're monitoring?
That is a great question, actually. At the moment, when we first started our program, thank you, George. When we first started our program, we were only focusing on the bay nettle jellyfish, but as we've gone through our first year of scrubbing, we've noticed that the lion's mane jellyfish also is a stinging jellyfish, also is native to this area. It kind of, this is a more southern part where the lion's mane kind of still lives, the habitat where they live. They're more northern and usually prefer colder temperatures. So we've been discussing monitoring lion's mane populations as well, as we've seen them more frequently, and people have been noticing them more this past season, which also just maybe because people tend to be living in their lagoon properties down here all year round, or more of the year than just the summer. They're using their homes not only as a summer vacation site now. So maybe there's just more people who are noticing that things that have always been there, but kind of hard to tell. So we're probably going to try to look into lion's mane jellyfish as well. Oh, and actually, now I'm thinking about it, not related to our project, but our jellyfish scientist, Dr. Bologna, also works on studying clinging jellyfish, which are very, very small jellyfish. They are in fact very invasive, and they have a very nasty sting. So at first the sting is pretty mild. You can notice it, but it's not that bad. But over time, it kind of stops your muscles from being, like, your muscles start to stop moving, and you have to be hospitalized, and you kind of just convulse in like you can't really move or do anything, which is really scary. It's not necessarily fatal, but if you get into a hospital in time, you should be fine. And it just takes a while to just get back to your normal healthy self. But those are invasive. They're from the more southern Atlantic Ocean. And they tend to cling onto eelgrass, and they're very, very small. They're about the size of a dime, so they're very hard to see until you're right next to them. That's not what we're focusing on for this project, but Dr. Bologna has been looking close at their population and their whereabouts in Barnegat Bay as they started to become more recognizable in this body of water.
Brittany, I have a question to kind of go off of that. Are the lion's mane and the clinging jellies, were they introduced here, or do you think it's because maybe waters are warming, or their range is changing and coming north? Just curious as how they got here.
So lion's mane jellyfish, from the research that I know, again, I'm not really a scientist, so I don't want to give wrong information, but what I've learned is that they do tend to be found in colder bodies of water, which is why they're kind of seen around this time right before summer hits. We've seen them a little bit while Barnegat Bay is still a little cool. They're native to the Atlantic Ocean, so and a little bit more north of us usually. So I don't think they're invasive from what I've learned. The clinging jellyfish are definitely invasive, and it's been suspected that because of warming waters, they've kind of migrated more north. They were also found, I think before they were found in Barnegat Bay, oddly enough, they were found a little bit more north in Massachusetts, maybe. I could be wrong. But in a couple of states a little bit farther north than us before they were found in Barnegat Bay, which is kind of weird how they kind of jumped and skipped over us. But now they're down in this area. And they usually die off, I think, once bayonetal jellyfish start to reach the adult maturity, they kind of see a decrease in the clinging jellies. So we suspect that maybe the bayonetal jellyfish kind of eat any of the clinging jellyfish that kind of stick around as the water gets warmer. But that's all, again, speculation. It's not proven yet.
Gotcha.
That's what we witnessed.
Okay, thank you.
Of course.
Any other questions for Brittany? All right, I don't see anything else.
Okay, I am just linking some links in the chat real quick. I'm also going to link the Berkeley Township Underwater Search and Rescue, their website. They're always looking for volunteers. If you're interested in diving, you don't have to be a certified diver. You don't even have to want to dive. There's plenty of things that they do that don't involve actually getting into the water. If you want to learn more about what they do and how you can get involved with them, I just want to link them too.
Yeah, great. Good to know. Thank you.
Of course. And then I will also just put my email in too, in case anyone wants to email me.
And I'll, after we conclude, I'll keep the room open a little bit, just give people time to copy and paste all that. Really, really great resources. So thank you.
Of course. Thank you.
All right. So I think it seems like there are no other questions. So we'll wrap things up. Thank you again, Brittany, so much for taking time out of your day later in the day too, to talk to us about this and learn a little bit more about what's going on with jellies in the Barnegat Bay. And thank you to the folks that joined us tonight. We appreciate you attending. And let me know if you have any questions about upcoming programs with the JC NERR. You can always reach me through email or phone. I'll put my email in the chat too. But other than that, we are all set for tonight. Thank you, everybody. Thanks for attending. Thanks, Hunter. And have a great rest of your evening, everybody.
Bye, everyone.
Yep.
Thank you all so much for having me and tuning in.
Absolutely. No problem. Thanks, Brittany.
Thank you.